Hi, everybody. It is me, and we're going to finish Chapter 7. This is Chapter 7, Section 5. This is fermentation and anaerobic respiration. So we just spent the whole last video lesson talking about what happens when we completely oxidize a molecule of glucose when oxygen is present. Step 1, glycolysis. Step 2, or 1.5, pyruvate moves into the mitochondria and is oxidized into acetyl-CoA. Acetyl-CoA enters the Krebs cycle, which is step 2. The Krebs cycle then is also called the citric acid cycle because the first product, when acetyl-CoA gets together with oxaloacetate, that's then oxidized throughout the cycle where we're pulling off all of these electrons and we are oxidizing the molecule and regenerating oxaloacetate. In the meantime, we are loading up our electron carriers, NAD, H and FADH too, and they are going to carry everything over to the ETC and unload their electrons there at the electron transport chain, which is oxidative phosphorylation and chemiosmosis, where the electrons go wee down the ETC, creating a proton gradient in the intermembrane space. They flow back into the matrix, and voila, we make gobs and gobs of ATP. But what happens if oxygen is not present? Because as you recall, oxygen is the final electron acceptor. It is the most electronegative item at the bottom of the ETC, and it's essentially pulling all the electrons down, and oxygen is reduced at the end of the ETC and becomes water and leaves the mitochondria. So what happens if there's no oxygen? Well, without oxygen, the electron transport chain will not operate. It is basically closed down. No ETC, no ATP, and at least not enough for us to survive, because I know some of you are going, well, Ms. Corcoran, I know ATP is generated during glycolysis and a little bit is generated during the Krebs cycle. That is true. So let's talk about how glycolysis couples with fermentation in a process of anaerobic respiration to produce ATP. Now, are we going to be able to survive on that? No way, because the yield during glycolysis is 2 ATP. And if you recall, our bodies, our cells need, 10 million ATP per second per cell? And every time there's a cycle of glycolysis, we get 2. So it certainly isn't going to keep us alive, but it can supplement us when we are in need. Okay, so there are a couple types of fermentation. Fermentation is basically glycolysis that happens in the cytoplasm, and it also includes reactions that regenerate our electron carrier NAD+, which can be reused by another episode of glycolysis. And 2 types of fermentation are alcoholic fermentation and lactic acid fermentation. In alcohol fermentation, organisms such as yeast, we obviously cannot undergo this because, like in the crash course video, if that were the case, every time we worked out, people would be drunk all the time and maybe the gyms would be really full. I don't know. I already like working out, and I think it would be kind of dangerous to be drunk when you do that. So not really a good choice. But let's go through this diagram. In the process of glycolysis, glucose is broken down, as you know, into pyruvic acid. It creates 2 molecules of ATP. It also reduces NAD+ into NADH. Perfect. That's not new information. However, if we want to do that again, we need to somehow unload. Remember, if we say NADH is our server and he's carrying electrons, he's standing there screaming, "What do you want me to do with all these electrons? What am I supposed to do with them? I have no place to put them. The ETC is closed." So in order to solve this problem so that we can break down another molecule of glucose, we have to get NAD+ back. We have to oxidize NADH. Normally, NADH is oxidized at the ETC. The ETC is closed. We can't unload the electrons there. So what happens is, in the particular case of alcoholic fermentation, is pyruvic acid actually loses a molecule of carbon dioxide, and then it gains the electron. So NADH is oxidized and acetaldehyde is reduced. And in unloading the electrons there, we create something called ethanol, and we also regenerate NAD+. So the whole purpose of fermentation is to regenerate NAD+ so we can oxidize glucose again into pyruvic acid. Now, once again, we're back in the same quandary with, oh my gosh, excuse me. I did not mean to do that. Oh my gosh. Sorry about that. We're in the same quandary where we've got NADH standing there going, what am I supposed to do with these electrons? The ETC is closed. What am I going to do? Well, once again, it's going to oxidize on acetaldehyde. You do not have to know this intermediate product. You just need to know that NADH is oxidized and we regenerate NAD plus. And in doing so, we have created some alcohol. Remember over here, we also create some carbon dioxide. So beer has bubbles. The bread, the yeast that creates bread, that causes bread to rise, goes through this process. The alcohol is made and baked off, but the carbon dioxide that's released causes your bread to actually rise. So here is a better and more up-close picture. Glucose is oxidized during glycolysis to pyruvic acid. We generate those two little molecules of ATP. We also reduce NAD plus into NADH. But we need to unload the electrons from NADH so we can do this again, because we can't do this again without NAD plus. In order to do that, pyruvic acid gets rid of carbon dioxide and accepts the electrons from NADH, creating ethanol and carbon dioxide gas. And we're regenerating the electron carrier NAD plus. The other type of fermentation is lactic acid fermentation. This is when pyruvate is reduced by NADH and NADH is oxidized, and we form something called lactate as an end product, or lactic acid, and there's no release of CO2. So let's look at this up close. Glucose turns into pyruvate. We know that during glycolysis. We make 2 ATP and we reduce NAD plus to NADH. Well, NADH then immediately gives the electrons to pyruvic acid, gives them to that, and it creates something called lactic acid or lactate. And in doing that, we have regenerated the electron carrier NAD+. You guys have all felt the product of this type of fermentation pathway. Human muscle cells use lactic acid fermentation to generate ATP when O2 is scarce. Okay, when our ATP demand is really high and our mitochondria cannot meet its need for ATP because we don't have enough oxygen, we will also supplementally undergo lactic acid fermentation. We could by no means live off of this, no way. But this explains sometimes why your muscles are sore after a particular exercise. So once again, the whole purpose of fermentation is to regenerate the electron carrier NAD+. The two different pathways have two different products. And luckily, they are named for their products. Lactic acid fermentation creates lactic acid. Alcoholic fermentation creates ethanol and carbon dioxide. Just remember, your bread rises and beer has bubbles. Okay, we can qualify or quantify, I guess we'd say qualify organisms as to whether or not they have to have oxygen or they don't have to have oxygen. An obligate anaerobe can only survive in anaerobic conditions. It cannot survive in the presence of oxygen. Anaerobe means no oxygen, and obligate means it's compelled or forced to do that. Yeast and many other bacteria, however, are what we call facultative anaerobes, which means they can survive using either fermentation or cellular respiration. We are not facultative anaerobes because we are aerobic creatures. We really need oxygen, or we cannot make the amount of ATP we need to survive. But if you're a facultative anaerobe, pyruvate is basically a fork in the road. And if oxygen is present, it'll undergo aerobic respiration. If oxygen is not present, then it will undergo anaerobic respiration. Okay, so let's talk about glycolysis. This is a pretty cool and fun fact. Glycolysis is the most common metabolic pathway among organisms on Earth. What does that indicate? Well, if everybody's got it, then it evolved a very, very, very long time ago, very early in the history of life. Okay, the last section of your book talks about glycolysis and the citric acid cycle and how it connects to many other metabolic pathways. We don't really need to know all of this information because you know how I like a picture way more than the words. So let's get to the picture. Basically, the whole gist of this is we've been talking about sugar, glucose specifically, as the model for what is oxidized in the catabolic processes and how we create the electron carriers for oxidative phosphorylation and making ATP. However, if you never, ever, ever eat carbohydrates, maybe you're a hunter-gatherer and you only eat the meat you kill and you don't ever eat any of the plants that are around or any berries or any seeds like that, and all you eat is meat, you are certainly going to be able to survive and make ATP through oxidative phosphorylation, because amino acids can be converted into several products along this metabolic pathway. Similarly, fats, some of the byproducts or the breakdown components of fats, glycerol and fatty acids, can enter this metabolic pathway and ultimately result in oxidative phosphorylation as well. We just sort of, as a rule in school, teach you that glucose is the main product that is used. Certainly that's the easiest one. But obviously, evolution would have not allowed us to only have one pathway. Or maybe there were organisms that only had one pathway. And what happened to them? They're dead. We don't talk about it anymore, because look at us now who have all of these pathways. We have survived and passed on the trait to have all of these pathways because it was more advantageous. Okay, so let's compare chemiosmosis in chloroplast to chemiosmosis in mitochondria. Both chloroplast and mitochondria generate ATP by chemiosmosis. Both the chloroplast and mitochondria create a proton gradient. They're going to fall down through ATP synthase. The mitochondria are going to use chemical energy from food. The chloroplast are going to use the light energy to create the proton gradient. Ultimately, both of them are going to rely on diffusion of protons down the concentration gradient through ATP synthase to drive the synthesis of ATP. This has a star in it. This is really, really important. This goes back to where you visualize those diagrams. You need to know the direction from which the protons are pumped and the direction that they fall back down into. In the mitochondria, protons are pumped into the inner membrane space and they fall back into the matrix. In the chloroplast, the protons are pumped into the thylakoid space from the stroma, and then they fall back into the stroma as they go through ATP synthase. And here is a really cool picture that is overlapped to both of these diagrams. So you've got, on the one hand, the matrix. We are pumping protons across the ETC as electrons go wee into the inner membrane space, and they're falling back into the matrix. If we look at this side, we're going to see we're pumping them from the stroma into the thylakoid space, and they're going to fall back out of the thylakoid into the stroma. Okay, just a super up close of that diagram. Okay, here's the review. As ever, I encourage you to go through these questions, and if you have any questions about the chapter, let me know.